Good afternoon, everyone. My name is Stephanie Espinosa, and I'm from Florida International University. Thank you for joining me today. My presentation is titled The Effects of Microplastics on Fertility Rates in Vertebrates. I'll be discussing how microplastics, or MPs, affect fertility across generations, with a focus on their endocrine disrupting properties and how these findings might reflect broader health implications. So let's get started. So, a brief introduction and a background in the issue of microplastics. Microplastics are tiny or minute particles of plastic, and they are less than 5 millimeters in size. They originate from the degradation of larger plastic objects or are manufactured as microbeads. These particles are ubiquitous in our environment. They're found everywhere. They're found in oceans, rivers, and even the air we breathe. Due to their small size, they're easily ingested by marine life and subsequently by humans through seafood, water, and other consumable items. MPs have been linked to a variety of health concerns, but in this study, I will focus on their specific effects on fertility. Specifically, the aim is to understand how MPs can disrupt reproductive health and the potential for multi-generational harm. In this first figure, it is called Methods of Accidental Inhalation of Microplastics Leading to Organ Damage. This figure visually introduces how microplastics can enter the body in a study named The Alarming Link Between Environmental Microplastics and Health Hazards with a Special Emphasis on Cancer. Microplastics, the problem in detail. Microplastics are not only a threat to the environment, but also to human and animal health. Due to their persistence, bioaccumulation is a major concern. Bioaccumulation is defined as the process by which an organism accumulates a chemical, such as a pollutant, in its tissues faster than it can be broken down or expelled. The organism accumulates and stores substances from its surroundings, such as water, air, and food, rather than discarding them. These particles accumulate in various tissues, including those of humans, and they can cause long-term damage. One area of particular concern is how microplastics interact with human tissues, potentially causing reproductive health issues. The bioaccumulation of microplastics in different organs suggests they have the potential to disrupt hormone regulation and reproductive health. In Figure 2, the increase in bioaccumulation of microplastics in various organs, including the human brain, demonstrates the widespread impact of microplastics on different organs and highlights their potential to disrupt hormonal and reproductive functions. This study does this by examining samples from various different studies from 2016 to 2024. It also connects dementia studies to microplastic accumulation studies in decedent brains. Research gaps and focus of the study. While there's a growing concern about the health impacts of microplastics, much of the existing research has focused on their environmental presence and general toxicity, as the last slide included. Few studies have addressed the long-term, multi-generational impact of microplastics on fertility, especially when considering the exposure across generations. My research focuses on filling this gap by exploring the specific effects of microplastics on fertility in vertebrates, with particular emphasis on endocrine disruption and reproductive health. Microplastics as endocrine disruptors. Microplastics are considered endocrine disrupting chemicals, or EDCs, due to their ability to mimic hormones and interfere with the endocrine system. One common example is bisphenol A, or BPA, a chemical often found in plastics that acts as an estrogen mimic. This disruption can lead to a variety of reproductive issues, including decreased fertility, irregular menstrual cycles, and lower sperm motility. In this slide, you will see two figures, Figure 3 and Figure 4. Figure 3, I have named endocrine disrupting compounds, EDCs, released by microplastics such as BPA. This figure illustrates how microplastics act as carriers for harmful chemicals like BPA, directly linking them to hormone disruption. In Figure 4, it is called the effect of nanoplastics and microplastics on sperm quality and testicular function in mice. This is an experimental study, and this visual helps reinforce the point about how microplastics negatively affect reproductive organs, specifically showing sperm abnormalities caused by microplastic exposure through photographs that you can see below. Moving on to reproductive health issues linked to microplastics. Exposure to microplastics has been shown to impact fertility across a range of species, including humans. Studies on mice have indicated that MP exposure can lead to decreased sperm motility, while in females, exposure is linked to irregular egg production and increased incidence of conditions like polycystic ovarian syndrome or PCOS. These impacts are indicative of the broader reproduction health risks associated with microplastic accumulation. In this slide, you will also see two figures, Figure 5 and Figure 6. Figure 5 is named PSMP accumulation in zebrafish tissues and the effect on reproductive health. This figure explains that polystyrene microplastics, or PSMPs, gather in zebrafish tissues in a manner that depends on concentration. Exposure influences the gonadosomatic index, or GSI, and the condition factor K, both used in this study. There are significant variations shown by error bars. This figure will visually demonstrate the reproductive effects in zebrafish, a commonly used model organism in science, showing how microplastics accumulate in tissues and disrupt reproductive functions. In Figure 6, you will see reproductive abnormalities in zebrafish, including gonadal issues resembling PCOS in female humans specifically. This is key for illustrating the long-term reproductive damage caused by microplastics, as it highlights how exposure leads to issues that resemble human reproductive disorders such as PCOS. This graphical abstract represents the findings of the study, concluding microplastics lead to apoptosis, oocyte maturation failure, and impaired steroid biosynthesis. Cancer risk and hormonal disruption. Beyond their direct impact on fertility, microplastics also pose a cancer risk. Microplastics can damage DNA and disrupt the hormonal balance that is crucial for cell regulation, leading to the development of cancers such as breast, ovarian, and prostate cancer. The role of MPs in cancer development is likely due to their ability to cause DNA mutations and interfere with the cellular processes that control reproduction and tissue regeneration. In Figure 7, Sources of Microplastics, Exposure Routes, and Health Hazards to Humans, it visually highlights how microplastics get into the body and pathways that lead to diseases, including cancer. This graphical abstract describes the sources of microplastics, the exposure routes and health hazards to humans, and the subcellular effects of plastic bioaccumulation. Mechanisms of reproductive health damage. The mechanisms through which microplastics damage reproductive health are complex, but can include hormone disruption, oxidative stress, and direct cellular damage. For example, in male mice, studies have shown that microplastic exposure leads to abnormal sperm morphology and motility. In females, ovarian health can be severely affected, resulting in irregular ovulation and the development of conditions like polycystic ovarian syndrome. In Figure 8, you see abnormal cavities in male sperm cells after exposure to microplastics. This figure is essential to show the direct damage to sperm cells caused by microplastic exposure, visually reinforcing the argument that microplastics interfere with male reproductive health. Abnormal cavities in male sperm cells in mice, black arrows indicate the abnormal cavities that you can see below, while white lines indicate normal cavities. Now we can talk about the multi-generational impact of microplastics on fertility. One of the most concerning aspects of microplastic exposure is its multi-generational impact. Studies have shown that not only are immediate effects on fertility noticeable, but these can be passed down to offspring, leading to a persistent cycle of reproductive issues. In aquatic species like zebrafish, even second-generation offspring exhibit reproductive impairments, which suggests that the damage is not just short-term, but long-lasting. In Figure 9, you can see developmental abnormalities and delayed hatching in zebrafish larvae exposed to microplastics. This visual is impactful in demonstrating how early life stages are affected by microplastics, showing that exposure in one generation can lead to multi-generational damage. This figure shows the tissue internalization of microplastics in the larvae using a magnifying technique. The sections that are identified as the eye and the muscles are stained with hematoxycline and eosin, and they show disorganization of the retinal pigment epithelial tissue, with microplastics indicated with arrows. Microplastics are shown among muscle fibers, and this figure shows malformations of larvae with PSMP exposure, polystyrene microplastics. Malformations seen include late hatching with microplastics on the chorion, spinal curvatures that are abnormal, edema, column deformation, and spinal curvature with blood pooling. Conclusions. In conclusion, microplastics are a serious environmental and public health concern. They disrupt reproductive health by acting as endocrine disruptors and causing direct damage to reproductive organs. The long-term, multi-generational effects are particularly alarming, and more research is needed to understand the full scope of the harm. Our findings suggest that actions should be taken to reduce plastic pollution, especially considering the potential for lasting health consequences. Figure 7 is shown again to summarize the environmental health risks of microplastic pollution and accumulation. Thank you for your attention, and I am happy to answer any questions.